Unit 4, lecture number 1, everything that you need to know about lecture 1, starting off with DNA replication. DNA replication, okay? So what do we need to know for this? Well, the first thing that we have to start off with is chromosomes and what they consist of, okay? So obviously, chromosomes, we know that they have something to do with DNA by now. But precisely what DNA consists of are DNA and proteins that are called histones. Let's look a little bit deeper at it, okay? So first of all, what you don't have to know is you don't have to know chromatin fibers. So none of this is important. But this should look very familiar. This is obviously a chromosome in a specific form that we're going to talk about later. And this is DNA, right? So entire chromosomes just spindled up DNA and little clumps of histones, which is the protein, alongside with the DNA. These are known as the nucleosomes here. So that's when you really zoom in on a chromosome. That's all it is. You do have to know histones, what histones are, and what kind of purpose they serve. Okay, the histones serve a role in the relaxation and the condensation of the chromosomes, which we're gonna talk about. So let's talk about that right now, actually. So relaxed, okay? We're going to just go over here. We're going to say relaxed. And then we have condensed. Relaxed and condensed chromosomes, okay? Have that about here. Condensed. All right, so let's do it like this. I'm going to use a different color. Let's use blue. That seems like a DNA-like color, okay? So relaxed literally just means a strand. So here in this picture, you can kind of see what I'm talking about. This is really strand-like, okay? This is an example of what a condensed would be, okay? So condensed basically means that this whole thing coils up and the histone to DNA strand ratio is really low, okay? So relax is just like this kind of single strand. A good example of condensed would be the same exact thing, but it would be shorter and fatter because basically this coils up. So these, you could think of these as little coils, okay? But one thing that's associated with condensed is something called supercoil, okay? So there's a clear kind of difference here between these two things. This is long and stringy, and this is condensed. The purpose that this serves is that this, and let's switch to a different color here real quick. This is the normal way that a chromosome is. Despite the fact that you've normally seen chromosomes like this that are condensed, that's because in the context of the majority of the things in biology, we look at them in the condensed form. But they're actually, most of the time, they're relaxed. So they're relaxed when not dividing. Okay. And I'm going to tell you why in just a second. So obviously, when if the chromosome is relaxed whenever we're not dividing, it's going to be condensed when it is dividing, right? So when it is dividing, we're going to be condensed, and this is for a specific reason. If you think about if you take a bunch of yarn and you cut it up into small kind of pieces, maybe about 3 inches long, and you put it in a big pile, right? And you mix them up. Let's say you mix them up in a Ziploc bag and you dump them onto your table, okay? And then take a bunch of dental floss and you cut those up into little pieces and you mix those up inside of a Ziploc bag and you put them on a table. Which one is going to be more easily taken apart strand by strand, right? It's going to be easier for the yarn because it's thicker, and it's going to be more difficult for like dental floss. I believe she uses an example like this in the lecture, but I'm getting right to the chase. And the reason why we're going to do that is you'll see later on in the next lecture when we talk about mitosis, these things need to be separated and placed into their own individual cells after they are after they're duplicated. Okay. Next concept, real quick, we need to talk about is the chromosome number. This is super, super simple. This is just the number of chromosomes in each cell of a species. Okay, I'm kind of shortening things down here because I'm writing with a mouse. So this means in every single cell, this is how many chromosomes are, minus a couple of exceptions, right? Ovum and sperm. But we're talking about all cells, right? So for humans, and this is going to be important, for humans, we have 46 chromosomes. This is our chromosome number. Okay, we have two sets of 23, making 46. This is our chromosome number. It's as simple as that. Chromosome number, number of chromosomes in the cell normally, and that is that. Next, real quick, we're going to talk about nucleotides. So you're going to have to know the three parts and the three structures that make up a nucleotide, which are sugar, and I'm going to get back to that in a second. So we have sugar, we have phosphate, and we have a nitrogenous base. Okay. Now, here's the thing. You're going to forget about the word sugar. This was in like Unit 1. You're going to forget about the word sugar. And the new term that you're going to have to know is deoxyribose. This is your substitute for the word sugar from now on. Forget about sugar, although it's going to come up again. So, show you an image of what this actually looks like put together really quick. As you can see, these are the three things that I previously mentioned, right? So we have the phosphate group, we have the nitrogenous base, and then we have a sugar. And right here you see deoxyribose. These are the three parts that make up a nucleotide, right? So we're talking about nucleotides here. I didn't really label this. These are nucleotides, and you've heard this before. You should already know kind of what these are. Okay, so this comes really important later, especially these negative signs. But I digress. Okay, so now we're going to zoom in and talk a lot more about these nitrogenous bases really quick. There are 4 different nitrogenous bases that we have to know. And I'm going to just put these in different colors just to be fun. Okay, so we're going to start off with thymine. Okay, we're going to go to, let's just go to orange, even though orange is ugly. Adenine. This should sound familiar from the last unit, Unit 3. Cytosine. I don't know why I didn't click that. Cytosine. And then finally we have guanine. Why does it do that? Guanine. Okay. Bottom line is we have to memorize these 4 things for our class. ATCG, thymine, adenine, guanine, and cytosine. So as soon as you have these 4 things down, this is just referring to the different types of nitrogenous bases. Now, if you look here, you will see in this particular example, you'll see 2 rings, right? We have a ring right here, and we have a ring right here. So nitrogenous bases can have 1 or 2 of these rings, and they are categorized as such. So these 4 things are categorized in their respective groups. So we have adenine and guanine. This is put into a group. We're going to talk about that. Okay. And then thymine and cytosine. This is going to be an entirely different group that we talk about. Just go over here. Okay. So let's start over here. Adenine and guanine, right? These are a part of a group known as purines. Purines. She gives a really good example in the lecture that I think is brilliant. And this is kind of a little trick to remember this. So again, we have adenine and guanine. And the way that she says is purines, pure as gold. Okay. This works. The reason why this is important is because all you need to memorize is the fact that you need to know all four of these, and you need to know that adenine and guanine go with the purines. And then you don't have to worry about this. I'm not even going to go over that. What I will tell you is that these are a part of a different group known as pyrimidines. Okay? Pyrimidines have one ring, and purines have two rings. So take a second and think about, would this be a purine or would this be a pyrimidine? Right, it would be a purine. You're going to have to know these. Again, you just have to memorize this, and the rest, you don't even have to bother memorizing. Just process of elimination for the rest. It's really not worth your time. Okay, these also will only pair with each other. What I mean by that is a purine can only match with a pyrimidine. So this kind of makes things convenient when we're trying to learn about base pairs. Just the same way that for purine and the pyrimidine, you only have to memorize this portion of it. For the base pairing, you only have to memorize half of it. Okay, so going down here, we are going to have A. I don't know why this doesn't change colors right away. We're going to have A, which is adenine. Okay, and it's going to pair with thymine. Okay, this is literally a word. Okay, this is all you have to remember. These two things pair with each other. That only leaves two other things that pair with each other, right? That's G and C, right? Guanine and cytosine. So really, only put this to memory and don't worry about it, but just know you're going to have to know what goes with what. And that brings me to the next point, which is single strand versus double strand, and then where we get the double helix name from. So these go with certain things, right? So let's say A, G, A, C, right? So this is a single strand of DNA. Okay. Now, what you're not seeing over here is there's a phosphate group and there's a sugar underneath here. But what the important thing that I'm trying to say is this is the nitrogenous groups, and this is what bonds to the other strand of DNA. Okay. This is a single strand, which we are going to work with single strands, but it's just really important to know. So A goes with T, G goes with C, and we have a T, and then we have a G. This is a second strand of DNA, and by the nitrogenous bases here, this is the thing that bonds with each other, okay? These bond with each other. They could be either double or triple. These are also, by the way, these are all bonded by H bonds. H bonds, okay? This is a double-stranded DNA. So this is part one of the double helix. So we have double helix, right? Everybody knows what that looks like. Double helix. This is the double. It's one strand here and one strand there, and these are bonded together. Now, the sugar is also bonding. The sugar is also bonding with the one right below it. So these are all tied together as well. These are all bonded together, and then we have these bonded together. This is a double strand. Now, the question is, where does the helix part come from, right? Why does DNA look like this right here and not just this, right? It's important to know that. This looks like a ladder. This is a double helix. So I just kind of flipped this over. So the words are a little weird, but that really doesn't matter. This is basically what DNA is, right? So we're going to have this nucleotide over here bonding, these bond with each other. Okay. Now, of course, one would be single and one would be double, but I digress. So these are also stacked on top of each other, right? So whenever you stack these on top of each other, why would they start to twist like that? And she mentions this in the lecture, and I'm glad that she, I'm really glad that she went over this because I've always just kind of been curious about it. So that's very simple, right? What we have here, these are all bonded to each other, and we have a phosphate group, and we have a phosphate group here. These are both very much electronegative. And what happens with things that are like that they have the same charge, right? They repel. Now, the thing about it is they can't really go anywhere vertically, right? These are bonded to each other, and this isn't strong enough to break a bond. So they, as far as separating vertically from each other, it's not really something that we can do. Okay, so we can't do that. The only choice that we have is that we twist. So this one would go, let's say this one would go in this direction, and this one would go in the other direction, and that would cause this to twist into that. The next part is really important. We have DNA replication, and there's two parts to the DNA replication. So the first part of DNA replication, okay, I want to show you a strand here. Right. So this is what DNA normally looks like, right? This is a single strand of nucleotides, double helix, right? The first step is to separate this so that we have some crap like this, and then maybe we have like a little loop. So a lot of these have been kind of taken off, and again, we have nucleotides here, but instead of being bonded all together, these are apart. To break these apart, this is DNA helicase. This is an enzyme that will break apart DNA, right? Enzyme ASE, Jesus. And this will break apart the DNA strands. The next thing that has to really happen, so this is really only two steps. So whatever. So this is part one. We're going to take this apart. The next part is we have another enzyme here, and this enzyme will do three things. First of all, this enzyme is called DNA polymerase. And this has three different functions. One, if we're going to copy DNA, we're going to say, okay, we have to read it, right? So number one, we have to read, and then we're going to get the complement or the pair. We're going to get the pair. How are we going to do that, and why do we need to read it, right? Previously mentioned above here, right? So if we have an A on one side, we know what other base pair to get to complete this. So if this is an A, we're going to get a T over here. And so this is a thymine nitrogenous base. And then this is the second part. So this right here, I shouldn't use this color. This right here is a nucleotide. This is the complement nucleotide that's gotten by DNA polymerase. There's a third thing that it's going to do also, which is going to be really important. It's going to be correct mistakes. Okay? It does all of these things at the same time. A really good way to remember this is that poly means many, and this is the only enzyme that we talk about that has multiple different functions. So again, we're going to read this. We're going to bring in the base pair. We're going to attach it. We're going to bring this. We're going to attach it. This is happening on both sides. We're bringing something in, we're attaching it. Bringing something in, we're attaching it. Bringing something in, we're going to attach it. So what happens is this is also happening at the same time. So this is going down on both directions. And then this is, she uses the term unzip. This is unzipping this into two separate strands. But at the same time that's happening, a brand new strand is being built on each side, if you can kind of see that through my messiness. What we're going to end up with, all right, is this. The copied part, the copied part, right? So we have one strand from the original going here, and we have another strand. Actually, let me use a different color because I feel like that's not a good idea. So we have one of these strands indicated by the black portion going here, and then we have the other strand that's going to go here. And then this part that was made by DNA polymerase is indicated by the blue portion of the double helix here. So this is another really important term. This is semi-conservative. Why? Because semi means half. So we are conserving half of the DNA strand with the replication, because half is here and half is right here, and the other half is copied. Now it's identical to it, but the actual original DNA is being conserved. Last part that we need to talk about is something that you already know about, centromeres. Okay, centromeres is basically what holds together two sister chromatids. So during DNA replication, a couple things happen. We start off with this. This is a chromosome, right? This is a shortened chromosome. It's short and it's fat, right? We're going to copy this, and we're going to now have another identical strand. Another identical strand. The tricky part about this is that before it was copied, this was, and let me go back, this is a chromosome, right? This is a chromosome. However, whenever we copy this, okay, this whole thing is, this whole thing is a chromosome. It's still a chromosome. So you have double helix. This is copied. Now we have two identical strands. This is still one singular chromosome. However, these individual parts, this is where it gets tricky because it wasn't explained. These two things individually called, these are individually called sister chromatids. Okay? In other words, once this is copied and we have two identical strands, this is not called a chromatid, and this is also not two chromosomes. This is still one chromosome, but it's two sister chromatids. This is why whenever she was going over the end of the lecture, and she talks about 46, everything's copied, and we have 46 chromosomes, but we have 92 chromatids. This will always be double the number of chromosomes. So that's the tricky part. This is still chromosome. And really quickly, a centromere is a protein right here that is used to attach spindle fibers during cell division, which is what this entire thing is about. So that is all that you need to know for the first lecture. So we're at about 25 minutes, a lot shorter than the actual lecture, a lot quicker pace, but you can go at your own pace, and that's all you need to know.